It's a pleasure to be here. And as you said, I'm going to talk about membranes and droplets. The membranes you already see here, but I'm going to introduce them with another slide. These are giant vesicles that I'm going to introduce. And we start with a textbook picture of the cell. If you throw out all the organelles out of the cell, and then you also remove the proteins, what you're going to end up with is a giant vesicle. These giants are relatively small, but they are gigantic compared to conventional vesicles with 100 nanometers in size. And they're a nice mimetic system, which represents the response of the membrane at the cell side scale. And they're also a very nice biomimetic compartment because you can now start reconstituting individual proteins, and you can reconstitute some cytoskeletal elements. And in this way, you can start building up cellular functionality, adding component by component. And this will be like a bottom-up approach for building what you want to do. Or you can also use the top-down approach where you force the cell to make you a giant vesicle. It's also called a bleb, or a giant plasma membrane vesicle, where all the plasma membrane proteins are properly reconstituted, oriented, and you have the whole zoo of lipids. You might be surprised to know that the mechanical properties of such giant plasma membrane vesicles are very similar to the mechanical properties of single or two component lipid vesicles, which is studied in these studies, which is to say that the underlying mechanics is set by the lipid bilayer. You can use these giant vesicles to measure properties, and this slide would review this very briefly. You can measure the stretching elasticity by aspirating giant vesicles in micropipettes, and you're going to get a number which is close to the stretching elasticity of a rubber sheet with the same thickness of 5 nanometers. The bending rigidity is measured in units of energy. 20 kBT is very small, which is to say that if several water molecules hit the membrane simultaneously, it would exhibit thermal noise, like shown here in this movie of the dancing vesicle. The shear surface viscosity you can measure by sticking a bead, or manipulate it with an optical tweezer, and then steering it along the membrane. And the number that you are going to get if you convert it into bulk viscosity would be around 1,000 times the viscosity of water. But still, movement of lipid species on the membrane is easy, simply because the membrane is thin and dissipation occurs into the environment. If you have such a setup, you can also pull tubes of very high curvature, cylindrical tubes from the membrane, and you can look at curvature preference of different proteins, for example, between the flat membrane and the highly curved one. And finally, if you stretch the membrane too much, you can create pores, and we look at the closure of such pores, which we induce with electroporation to measure the edge tension of the membrane. So if you got interested in all of this, here is a book for you, a giant book about giant vesicles, which is a kind of a textbook-like material, which introduces various protocols and approaches about using membranes, giant vesicles, to learn more about membranes. There are many labs that use giant vesicles, and people typically look for the ideal or clean vesicle, but very often one observes tubes and buds, and the tubes can be inward or outward. They can be very thin, or they can be pull chain-like, and they mean something. They mean that the membrane prefers to take or adopt a certain curvature, which is called spontaneous curvature. It describes the bilayer asymmetry, which can be as a result of a different composition of the two leaflets, or different composition of the solutions across the membrane, symmetric ion or protein adsorption. The nomenclature is such that the membrane bends outward, and then the curvature is positive, or if it's negative, it's the membrane bends inward. And this is one example of small positive curvature. So the bud here that is produced is large, so this is small curvature. It's outward, that's why it's positive, and here it's negative, high negative curvature. The message here is that just looking at these giant vesicles morphology, you have a kind of a direct and fine sensor for membrane asymmetry. And now I'm going to give very briefly two examples of such tubes and asymmetries. The first one is GM1, a sugar-like lipid, which is predominantly situated at the outer leaflet of the neuronal membranes. And this was work initiated by Nico Fricke and Tripta Bhatia, who prepared vesicles that had more GM1 inside, which generated negative spontaneous curvature in a lot of tubes. These tubes are connected to the vesicle body because, and we know that because if you start aspirating these vesicles, the tubes are reabsorbed to the surface. And if you release the tension, they would reform. We also see that up to 50% of the vesicle area can be stored in such tubes, which is to say that cells can also use a similar mechanism of storing area that is needed afterwards for protrusion. And this area can be stored in such tubes simply, rather than fusing vesicles and producing excess area for protrusions. Now, to measure the spontaneous curvature, and this is work initiated by Arakti and Dasgupta, you can measure the force with an optical tweezer, which is needed to pull a tube. This force is proportional to the tension of the membrane, which you set with the pipette, and you can measure with this aspiration pipette. Kappa is the bending rigidity, and then you have a term which reflects the spontaneous curvature M. Now, if you take a membrane which is purely symmetric, just made out of one lipid called POPC, you look at the black data, the slope would give you the bending rigidity and the intercept would give you the spontaneous curvature. This is a symmetric membrane, so the spontaneous curvature is 0 and the slope is 0. If you start doping the membrane asymmetrically with this molecule, you start getting some non-zero intercept, and you can get what is the spontaneous curvature out of this intercept, which is of the order of 1/100 to 1/200 nanometers. Now, we were then wondering, okay, so asymmetric adsorption of molecules can drive curvature. How about ions? And this is motivated by the asymmetry across the plasma membrane. Outside, you have a lot of sodium and chlorine ions. Inside, you have potassium ions, but no chlorine ions, and the electron neutrality is balanced with macromolecules. So we tried to develop two systems that mimic this kind of asymmetry, and this was work initiated by Marzi Karimi. So in the first system, we have low asymmetry, just a little bit of sugar and a little bit of salt outside, both isotonic, like osmotically balanced. And compare this to a highly asymmetric system where you have a lot of sucrose inside and osmotically balanced salt outside. So Marzi did the same experiment, measured the force as a function of tension and looked at the intercept. And for the low asymmetry system, the intercept was negligible, reflecting negligible spontaneous curvature, while here, just by this asymmetry of salt and sugar, you get this high negative spontaneous curvature, which is why when we deflate these vesicles, they produce these tubes. And because the spontaneous curvature is inversely proportional to the tube radius, we know that these tubes have a diameter of 1/100, 1/120. Now, so there are various asymmetries that can cause shapes in the membranes, and we were wondering about the crowded cytoplasm. Here's a movie illustrating how crowded this environment is. So you have a lot of macromolecules, and if you think about that, the Gibbs phase rule would tell you that in such a crowded environment, you should have different phases. And this is the one illustration of this in the cell for P granules. So these are droplets or phases, liquid-liquid phase separation. And some of you might be aware that this is now a big hype among the biologist community, talking about liquid-liquid phase separation, biomolecular condensates, or protein-rich droplets, all of this. If you look into the literature already in the 80s, this has been known. Now we are rediscovering the wheel more or less. But if you look at such electron microscopy data, you are going to see that these protein bodies called before are actually droplets because they are smoothly curved and they can deform the membrane. So this is what we were wondering. If you have so many droplets and you have so many membrane structures inside the cell, what happens when the droplet meets the membrane? And to study this, and this is work that we initiated already quite some time ago, almost 15 years ago, with Yonghang Li and Yonggang Liu, we mimic the crowded environment in the cell by encapsulating in our kind of artificial cells. We encapsulated macromolecules like PEG and dextran. Polyethylene glycol and dextran are simple molecules, well known. They are water soluble. If you mix them, you are going to get an aqueous solution. But if you increase the concentration of PEG and dextran, you're going to end up with phase separation in your test tube where you have two aqueous phases. The dextran-rich one is heavy and it's at the bottom, and the PEG-rich phase is at the top. So we wanted to have this phase separation inside the vesicles by crossing this binodal. And you can do that by exposing the vesicles to high osmolarity solution. The membrane is permeable to water, so water leaks out and the polymer gets concentrated, and there you go, you have phase separation inside. This is a vertical cross-section of the vesicles, and here is a phase contrast of the process, which is sped up. It takes about 2 hours. Here you see the condensation of the dextran-rich phase, and the faint contour of the membrane is here, if you see my pointer. Now, in these conditions, the dextran-rich droplet does not want to be in contact with the membrane, but if you go further, tada, there is a wetting transition. Now the dextran-rich droplet partially wets the membrane. There's an exclamation mark here, even though wetting transitions have been known for ages about droplets on solid substrate. The interesting thing here is that the membrane is not a solid substrate. It's a very soft substrate. And you can imagine that if the interfacial tension here is very high, it should be able to deform the membrane. And this is what we see here. With further deflation, the dextran-rich phase here starts protruding out. The membrane goes around to enwrap both of them, which we see also with confocal microscopy. And further deflation can even lead to separating the vesicle into two compartments with two different solutions. If the phase separation occurs outside, you can end up with droplets wetting the vesicles and having such moon-like shapes, moon-like shape vesicles. And if you have enough area, you can you can even have engulfment or endocytic processes where the vesicle wraps the whole droplet. When you have too much membrane, it gets stored into tubes that accumulate at the interface. And here is also a vertical cross-section of a vesicle with the dextran-rich droplet and the PEG-rich one here, and the tubes accumulating here. If you look from above, you're going to see the spaghetti-like structure of membrane nanotubes adsorbed at the interface simply to minimize the interfacial energy. The tubes are stabilized by spontaneous curvature generated by the weak adsorption of PEG on the membrane, which you know from simulations that use exactly the membrane composition that we have. When the interface gets very crowded, we see that we don't see anymore much with confocal images, but work initiated by Ziliang Cao using STED microscopy, super resolution microscopy, we could resolve these nanotubes at the interface. What Ziliang also observed is that at the right interfacial tension and excess area, these tubes that are at the interface can start transforming into double membrane sheets. So just watch the end of this tube, which transforms into something that looks like a balloon. This is actually a very flat balloon, or we call it a double membrane sheet or double membrane, very flat, pancake-like, looking like a vesicle, which is reminiscent to the organelle shapes of the cisterna-like structures of the endoplasmic reticulum. Here are more images of this, and this is just a projection in the vertical direction just to show you that these double membrane sheets are very thin. Now, okay, you can say, fine, PEG and dextran can do all this, so what? What is the relevance to biological crowding in systems? So coming back to this slide, again, these are protein-rich droplets, and we also looked at protein-rich droplets, and this was work initiated by Nanan Chen, who used soy glycinin. This is a major storage protein in the vacuole of plant cells of soybean seeds, And now looked at what happens to glycine in solutions when you change salt, pH, temperature. And she could kind of resolve the phase diagram of this system, finding a region in the phase diagram where condensates form or droplets form, protein-rich droplets. And then Agustin Mangiarotti in the group brought these condensates in the vicinity of giant vesicles and look at how they interact. At low salt concentration, they don't seem to like each other, but if you increase the salt, or if you can also play with the membrane charge, you can modulate that and go from complete deweighting to partial weighting, and to even complete weighting when the droplets want to fully spread on the surface. The stickiness is characterized, you can think of a contact angle, but this is true only if you have a flat surface. If you have a membrane that bends and makes something that looks like a kink, this contact angle is not sufficient to characterize this system, because as you see here, the same system, the same membrane composition, same droplets exhibit different contact angles. I'll skip this slide where I wanted to tell you that there is a way to go around and introduce a geometric factor that can describe this, and it's a material property contrary to the contact angles that one can optically measure. And go to tell you that when you have wetting, when the droplets want to wet the membrane, when there are such conditions, we can observe that vesicles can breach condensates, or condensates can breach or like connect vesicles, or you can think of role of condensates as a connectivity mean between membrane-bound organelles in the cell. If you have these droplets, you know that in the cell very often conditions change locally. So if you just have an upshift or downshift in salt or pH, you can induce phase separation inside these droplets, ending up with something called hollow condensates. They still have aqueous solution inside, but the shape is of a hollow structure. They have the same affinity to the membrane as the normal condensates, but it looks as if hollow condensates could provide additional means for compartmentation in cells, where you can think of one reaction, the cell focusing one reaction in one compartment, or the protein-rich droplet, or the membrane organelle. What condensates do to the membrane when they wet them is that they slow down the diffusion in the lipids. So here we have fluorescence recovery after photobleaching experiments done on a vesicle that on one side has a droplet wetting it. So in the wetted part, we see that the diffusion is twice slower compared to the bare part of the membrane. Using environmentally sensitive probes such as Laudan, we also resolve that the packing in this wetted region in the membrane is increased and the lipid gets dehydrated. At the same time, we also see that both the vesicles and the condensate mutually mold each other. If you start with such a system and you deflate the membrane, you see that it starts accumulating at the interface, and then it forms protrusions or ruffles. They are even more spectacular if you have more excess area. And this is an interesting picture here showing that the flat part of the membrane, so this is basically flat on molecular level, coexists with a very wiggled surface or protrusions on the other side of the vesicle. If you apply some tension on the bare part of the membrane, you can pull these wiggles out, and they would reform if you, the ruffles would reform if you release the tension. So playing with the area can be done not only with deflating such vesicles, but one could also use photoswitchable molecules that can influence the area and the tension of the membrane. And this is one such molecule, a lipid, that undergoes conformational change with UV and blue light, which is reversible. And this was work initiated by Mina Alexanian in the group. So when you shine light to such vesicles, they increase in area, and then you can shine blue light and this reverses. And this is one movie here showing a very special vesicle that when you shine the UV light, it undergoes this transformation, which is completely reversible when you shine the blue light. So why am I telling you about these membranes? Coming back to the droplets, we were wondering if we have this regulator for membrane area, whether we can influence to what extent the membrane wraps the membrane wraps the condensate. And it turned out that this is feasible. So you start with a membrane sticking to a condensate, but then when you shine the UV light and increase the area of the vesicle, it tries to wrap more or eat more of the condensate. And this is a movie showing this. This is reversible with blue light. And if you have smaller droplet and a larger vesicle, you can even go to the extent of complete engulfment, or something like you can think of endocytosis, but this is reversible. So you shine blue light and the droplet goes out. And the final thing I wanted to tell you about is that so condensates can do much more than wet, bridge, and deform membranes. They can also stabilize vesicles upon damage. And this was work in collaboration with the Gutierrez lab in the Crick Institute in the UK, where we did the following. So now we encapsulated our protein in a homogeneous state. So we are coming back to the phase diagram here. We start somewhere here, where you have some homogeneous solution of the protein, no phase separation. And this is our vesicle. And outside, we exchange the solution such that if the protein leaves the membrane, it would end up in this region where condensate should form. At the same time, we create a pore by using a hypotonic shock. And when the protein leaves the membrane, it forms the condensate, which localizes at the pore. The way we do that is by,
Rumi, just to let you know, you have about maybe 4 or 5 minutes.
Yes, I have 2 more slides. That's great.
Perfect.
Thank you. So the way we do that is we have these microfluidic chips where we trap our vesicles. So these are PDMS posts, and then we flush the solution that we are interested in, and then we look at certain vesicles. So here is a movie. These are the microfluidic posts and our vesicle, which initially had this glycine solution, homogeneous, but then when it porates, the condensate forms. Everything is very dynamic because we have a flow and the whole thing turns around. But if we stop the flow and do some imaging, we see the formation of the pore. The protein leaks out, and the moment it meets the solution outside, which primes it for condensation, the condensate forms, and at the end plucks this or patches this pore, and then you have this moon-like shape looking vesicles with the craters on them plugged by the condensates. Without the condensate, such vesicles, so these are just 2-component membranes with some charged lipids. If you porate them, they would typically burst. And here's some movies that I like showing of porating and bursting vesicles, but they're irrelevant now for this presentation. I just like this movie. That's why I'm showing them. A more relevant system that we looked in, again, in a similar way, was an attempt to kind of reconstitute stress granules and look at their membrane damage stabilization. And the motivation was that the Gutierrez lab has observed some colocalization of stress granules with lysosomes that are damaged. So these stress granules, one can reconstitute them in vitro by using this protein G3BP and RNA. This is collaboration with the Franzmann lab. So what we tried to do with our giant vesicles is to build an inside-out lysosome. So typically in lysosomes, the pH is low. In our case, the pH was, we reduced the pH by outside. That's why we call it inside-out. Inside, we encapsulated G3BP1, and outside, we added the RNA. When these 2 molecules meet in the right pH, they would form a condensate. And at the same time, we porated the membrane and look whether condensates would form and stabilize the membranes. And this is indeed what happened. This movie is a little bit more complex, but over time, one sees that after poration, the condensate forms, and then if you do some imaging of that, you see the pore in the membrane that is plugged by the condensate. So I come to my conclusions. Membranes don't need much. A little asymmetry or droplet wetting is enough to trigger tube sprouting, budding, engulfment, and even repair and fission. I did not talk about fission, but then isn't it obvious that the shaping of organelles such as the endoplasmic reticulum or Golgi or the mitochondria can be driven not only by protein functionality, but simply also helped by physicochemical factors such as spontaneous curvature generation, wetting, and phase separation. And these are the people that I already mentioned in the talk. I'll just say that we're setting up a network, come and sell on condensate membrane scaffold. So we'll have 17 open positions for PhD students. You can contact me about that. And I'll stop here. And thank you for your attention. I'll be looking forward to your questions.